Hi guys, we're back, and this time we are looking at hormonal communication. Remember, this is a revision video, so we might not get into every single little detail, but broadly speaking, we're going to try and put all the content that you need to know on this board so that nothing can escape your revision process. Okay, so with hormonal communication, I'm looking at the specification right here. There's three main parts to hormonal communication, and it is in many ways it's a counterpoint to nervous communication or neuronal communication. And we are going to touch a little bit on that, but broadly speaking, hormonal communication is about how hormones work. So generally speaking, how do hormones allow communication between different and distant parts of the body, so that there is that, and there's a little bit of contrasting with neuronal communication. Then, as our two examples of hormonal communication, we have the adrenal glands, which really they're inserted, so this is inserted here, but really the adrenal glands functionally belong to plant and animal responses, because we functionally look at adrenal glands when we look at the stress response and maybe a little bit in glucose homeostasis, but also in osmoregulation. That's to do with the kidneys. So that's in homeostasis as well. And then as our other example, we have the pancreas. So how hormones work, adrenal glands as an example of endocrine glands, and pancreas as another example of endocrine tissues. Okay, so there we have those three areas. Let's get into it. Okay, so in terms of how hormones work, so hormones are produced, right? Hormones are chemicals that are produced by the cells of endocrine organs. So we have their story begins with the endocrine organ. All right, and from the endocrine organ, the hormone will then be secreted directly into the blood. So that's different from the nervous or neuronal communication. Remember, neuronal communication is an electrical signal. This is a chemical. The signal travels via cells, via neurons in neuronal communication, whereas this is in blood. But anyway, let's not get too much into that. But once in the blood, remember that hormone will then find its target, or it will penetrate to all parts of the body, and it will find its target cells / tissue or / organ, right? And there it will bind to a particular receptor, complementary protein receptor. And remember, target cells have the receptor, whereas cells which are not targets are not supposed to respond to that hormone. They would not have a complementary receptor, and so that hormone would just kind of bounce off of them completely. Okay, so we have a receptor, and as a result of binding the receptor, we would get cell signaling, and that cell signaling would then result in the cell behaving differently. Maybe a new protein is produced, maybe certain proteins are activated, certain other proteins are deactivated, but the cell would behave differently as a result of binding the hormone, and we would get some response. Okay, and as we look at the adrenal glands and pancreas, we'll look at examples of that. What's going on with my voice today? All right, so that's the endocrine, generally the summary version of how hormones work, we are going to go in a little bit more depth into that, though, however, because as hormones, the different types of hormones can also, like, we need to be aware of the different types of hormones. Okay, so generally speaking, there are two major classes of hormones. One are, or the first kind, or one kind, is peptide hormones, and they're basically amino acids joined together. There is a gene for them in the chromosomal DNA, and just like any other protein, they get transcribed, translated, modified, packaged, and released out of the cell. So we need to be aware of this kind of hormone. Okay, now these generally tend to be hydrophilic. Okay, and they are water soluble, and so they can't cross the cell boundary, the cell membrane, and so they can't really penetrate into the cell. Okay, and because of that, when they bind a receptor on the outside of the cell, so they need a cell surface receptor. Now, these are the basics. I'm just going to go into a bit more detail here. Okay, they have a cell surface receptor, and as a result of the cell surface receptor being engaged by the complementary hormone, second messenger will be produced inside the cell, because the first messenger, the peptide hormone, can't get inside. So the binding of the hormone at a cell surface receptor will trigger events inside the cell, leading to the production of a second messenger. Okay, second messenger, and the second messenger will then cause cell signaling events. Maybe the second messenger binds to a protein and activates it. So in the absence of the hormone, that protein was inactive, but now that the hormone is present, that protein is now active, and certain things will happen causing a response. Okay, because later what we're going to discuss is hormone is released, response is initiated. So basically what we're setting out is a kind of vague general mechanism as to how hormones can work, so that later it's not like a black box, because hormones are going to cause responses, and we're not really going to be too concerned about how those things happen. But we do need to be aware of hormones, where they are produced, the effects that they have, i.e. the responses that they produce, and we need to have a general idea about how those things could happen, though we don't need to know the specifics of how those things happen. So cell signaling and etc. response. Okay, right, now the other type is steroid hormones or lipophilic hormones. Let's just call them steroid hormones. Now, steroid hormones are hydrophobic or lipophilic, and as a result, they can just diffuse across the cell membrane because they can interact with the hydrophobic fatty acid tails of cell membranes, and they can go right through. So their receptor is cytoplasmic. So they go right across the cell membrane, and they have cytoplasmic complementary receptor proteins. Okay, and so what happens as a result of the hormone binding the receptor in the cytoplasm? What's different? Well, once these proteins bind the hormone in the cytoplasm, they usually travel to the nucleus and activate or alter or act as transcription factors, basically. Okay, so they go in the nucleus. and the hormone binding to the protein changes its shape, therefore changes its function. So before it couldn't act as a transcription factor, but now it can act as a transcription factor, and it goes into the nucleus, acts as a transcription factor, and alters gene expression. So whereas with a peptide hormone, we were altering protein activity, generally speaking, as a result of the second messenger, in the steroid hormone, we're not just simply activating proteins, we're actually getting proteins to be produced. We're initiating transcription, translation processes to make new proteins, and therefore with the creation of these new proteins, or with the production of these new proteins, then we will have new cell behaviors which will result in responses. Okay, so this is generally speaking what we're talking about in relation to hormones. Okay, I think there was some other stuff to do with hormones. I guess what we could do here, which I'm not going to go into right now, so I think I've already gone into too much detail, is this idea of contrasting with neuronal communication. I think this is an important part of this. Now, yes, this year the neuronal communication technically is not on the advanced information list, so there is that. However, plant and animal responses is there, and there is an aspect in there of neuronal communication built into that topic. So I think it's worth including this as part of your work. Okay, so like, for example, okay, I'm not going to go into detail because honestly I don't remember them all right now. Okay, but we're thinking about things like speed, which acts more quickly, right? Neuronal communication, electrical signals happen very quickly. The production of a chemical from the cell, its release into the blood, its movement through the body, and then diffusing through the tissues, and then initiating, the signaling, and so all those things could take time. How widespread? So is it specific, or can it have wide-reaching effects, or is it very, very targeted? Like, does it have lots of effectors, or does it have one or just a few specific effectors? Okay. What else could there be? There's speed, there's specificity, there is the nature of the communication. One is chemical in nature, one is essentially electrical in nature. So, I mean, and there's other ones which we could compare, but the point I'm trying to make is have that list of similarities and differences. Okay, right, and then we get onto the adrenal glands, but then the main business is going to be to do with the pancreas and glucose homeostasis. Okay, so then for our first example we go to the adrenal glands, and if you don't mind, I will expand a little bit beyond hormonal communication just to put the stuff on plant and animal responses a bit into context, in the context of hormonal communication, just to make it clear how the two connect. Okay, so the adrenal glands, what we'll begin with is the structure of the adrenal glands, and it's not too complicated. It's a kind of pyramid shaped organ that's sitting atop the kidneys. Okay, And the thing to remember structurally is that there's an inside bit called the medulla and an outside bit called the cortex. Okay, so the outside bit is called the cortex and the inside bit is called the medulla. Medulla, medulla. Okay, and we need to have an awareness that these two parts produce different hormones, but also get stimulated to produce and release those hormones in different ways. All right, and I always forget about this, so I'm going to refer to the textbook at this point. Okay, so the cortex produces three types of hormones. Okay, the first one is glucocorticoids. I'll leave myself enough space here. The first one is... Okay, so we have glucocorticoids. These control the metabolism okay, and affect blood glucose levels. Now, what's interesting here is, as part of the stress response, cortisol is a glucocorticoid. Okay, so I'll just put it here. Cortisol is an example of a glucocorticoid. It affects the metabolism such that it increases blood glucose levels during the stress response. Okay, so cortisol, it affects liver and muscles, right, and it increases blood glucose. So that's important right there as part of the stress response. Other hormones also produced by the cortex are mineralocorticoids, which affect the blood water levels, salt concentration in the blood, and we have androgens. These are sex hormones. Okay, so we just need to know of these. Okay, right. Now, the other important thing about the cortex is that it is stimulated by hormones. It's stimulated by other hormones in the blood. Okay, so all of these hormones are produced by the cortex when the cortex itself, which is a target of other hormones, gets hormonal signals from the pituitary gland. Okay, so the cortex is stimulated by, we can call them pituitary hormones, but we can say stimulated by other hormones. So as part of the stress response, the stress in the brain resulted in the hypothalamus releasing a hormone that affected the pituitary gland. The pituitary gland then released a hormone that went around in the blood, arrived at the adrenal cortex and stimulated the release of cortisol. So that puts that puts that story into context here. Let's go over to the medulla then. The medulla is actually stimulated by sympathetic motor neurons. Okay? So the medulla part is not stimulated by other hormones, it is stimulated by sympathetic motor neurons. Okay, and when this happens, when the impulses come down the sympathetic motor neurons, the medulla releases steroid hormones, adrenaline, and noradrenaline. A quick correction, the adrenal medulla releases catecholamines, not steroid hormones. Okay, and again, we know the effect that these have on the stress response, stimulating the heart rate or causing the sinoatrial node, the rhythm of the sinoatrial node to increase, or the rate of contractions from the sinoatrial node to increase. Okay, so I hope we understand how what we discussed in the plant and animal responses in terms of the stress response, how that ties in here to this topic. Okay, and this is I guess this is an ideal thing that examiners would perhaps take advantage of, the cross-pollination between two topics to ask their questions. The cortisol targets the liver and causes an increase in blood glucose levels because it stimulates gluconeogenesis, glycogenolysis in the liver. Similarly, adrenaline also has that effect. So adrenaline affects the liver and causes blood glucose increase. Remember though that the main target of adrenaline is going to be the sinoatrial node and an increase in heart rate. And noradrenaline causes vasoconstriction to the non-essential systems in the body during a fight or flight response. Okay, so kind of diverting the blood flow, less to the digestive system, for example, and more towards the muscles. Okay, so we'll just say here vasoconstriction from a point of view of, right, let's get the blood to where it's needed more. Now the pancreas has two kind of main types of cells that we need to be aware of. So if we're looking at the pancreas, we're really concerned. If we zoomed right into a little part of the pancreas, what we'd see is just kind of lots and lots of cells, but those cells are kind of grouped according to their function, because remember the pancreas has two functions. So there's some cells which are called the pancreatic acinar cells, which I don't know if I've pronounced that correctly, or acinar cells, which essentially what they do is they open into a duct. So there's an opening here, and these cells, which will be secreting digestive enzymes, right? So these cells will go through the process of transcription, translation, protein modification, exocytosis, and through that process they will be releasing enzymes into what we know as pancreatic juice. So, and then these spaces are known, that's these will be dotted all over. Okay, so these pancreatic acinar cells will be all over the pancreas, and each of those little openings, these ducts, they'll come together and they'll converge to form what is known as the pancreatic duct. So that's the pancreatic kind of duct essentially, and these are the acinar cells. This relates to the role of the pancreas as an exocrine organ. Okay, exocrine. One of its jobs is to produce the digestive enzymes via the pancreatic duct to the small intestines or the duodenum, and those enzymes mix with the mashed up food from the stomach. They carry out the hydrolytic reactions required to break the large molecules into small molecules, which can then be absorbed. So if we zoomed in and had another look, we might find that there's other groups of cells as well. All right, and in these islets of Langerhans, there are two types of cells. There are the alpha cells and they are the beta cells. I have a problem writing S's. So the beta cells produce insulin, okay, And the alpha cells produce a hormone called glucagon. Both are part of this negative feedback idea. So if we had like a little graph here, and let's say that this line was the set point norm, when blood glucose levels rise above the set point norm, insulin is produced, okay, and released, and this causes tissues to absorb glucose and remove glucose from the blood, which causes the blood glucose levels to decrease. When it gets too low, insulin production stops, glucagon production and release increases, and blood glucose levels rise again, and so we continue. What is it that insulin is doing? So what are the effects of insulin? Okay, and what are the effects of glucagon? It's usually the reverse. Okay, and we do need to spend a little bit of time on how the beta cells detect the high blood glucose level and release insulin in response. Okay, so insulin, beta cells, insulin, actually it's not about production, it's more about release. What are the effects of insulin? Right, increased transport into cells. Other insulin effects include increased rate of respiration. So as the respiration rate increases, we're using up more of the glucose in the cell, in the cytoplasm, which lowers the cytoplasm concentration, which allows more glucose to come in to the cell, perhaps via facilitated diffusion. Right, we have increased glycogenesis. Glycogenesis, we are creating glycogen. We're either saying to the glucose, right, get used up in respiration, or get converted into the insoluble glycogen, right? So that doesn't count as glucose. Right, we are increasing glucose to fat conversion, increasing the glucose to fat conversion. An important one which I always forget about is that insulin inhibits glucagon production, and that's the kind of the crux of the whole antagonistic relationship between the two, right? Inhibits. So while glucagon levels are high, while insulin levels are high, glucagon levels are kept low, right? Inhibits glucagon production or release. The major targets of insulin are the liver and skeletal muscle, but there's wide range, like a lot of the body's tissues have insulin receptors and can respond. Okay, right. Glucagon will essentially, it will do the opposite. It will act to increase the blood glucose levels. So for example, effects will include, it will carry out glycogenolysis, or it will stimulate glycogenolysis. So the breakdown of glycogen into glucose, so that cell concentration of glucose increases. Glucose then diffuses out of the cell, then into the blood, where the blood glucose concentration can increase. Glycogenolysis. Increased glycogenolysis, reduced glucose absorption, increase gluconeogenesis. Remember, that is the conversion of things that are not glucose into glucose. So using, for example, amino acids to make glucose. That's happening in the liver. And once the liver converts amino acids into glucose, it will release that into the blood. Oh yeah, there is the mechanism of the insulin release. I'm not going to go into it. It's there. So release. I'm just going to put it down here. Know how blood glucose, know the sequence of events where increased blood glucose, what are the sequence of events that results in increased blood insulin levels. Okay, it's 4 or 5 points, it's not that deep, so just know that part. So there is that, and then we have diabetes, and I think I haven't left myself enough space. In terms of diabetes, that there are 2 types. Okay, so know the types of diabetes. So there is type 1, which essentially is a reduced production capacity of insulin. It's because we have a loss of beta cells. Okay, so because of a loss of beta cells, it might be because of an autoimmune process. We've got a loss of beta cells, therefore we don't have the capacity to produce as much insulin, and so we're not able to keep the blood glucose levels in control. Type 2 is a result of, if blood glucose levels are consistently very, very high, if insulin levels are therefore high a lot, over time there could be a reduced responsiveness to insulin because it's always there. And as a result, there's a reduced responsiveness to insulin, but the end result is the same, that the blood glucose levels are too high. The last bit is about treatments. Okay, so in terms of type 1, the treatment option or the treatment is insulin injection. I would have a few arguments. What are the practicalities of this? What could go wrong with this? So positives and negatives. Type 2, we could have diet control. So at the earliest levels, we could have diet control, lifestyle adjustment. Okay, but then if that doesn't control it, we could have drug treatments. So drugs that can control or influence the blood glucose levels. And finally, if it gets even more serious, even type 2, we might need to resort to insulin injection. The insulin that we use in the injection can be derived from different sources. Initially, there was the animal derived insulin, which could have people could have allergic reactions to that, might have ethical objections to that, religious objections to that. As an alternative, genetically modified bacteria could also then be used to produce the insulin. So the gene for insulin could be inserted into a vector piece of DNA, and the vector DNA it used to transform bacterial cells. Those bacterial cells then translate, essentially express the human insulin gene, and the insulin produced could be separated, purified away from the bacteria, and used to treat humans. Now, without all the disadvantages of using the animal-derived insulin. Okay, so we have that, but also we have stem cells potentially. Okay, so stem cells are, well, this is still in kind of, stem cells have the potential to become any kind of cell, right? They have the developmental potential to develop or differentiate into potentially beta cells, which, remember, in type 1 diabetes, or the number of beta cells has decreased, reducing our insulin production capacity. Now, if stem cells could be directed to or stimulated to differentiate and into beta cells, this could be a way to restore the insulin producing capacity of a person that's suffering from type 1 diabetes. So there is that. Okay, and remember in module 2 we do look at stem cells, and we do look at the positives and negatives of the use of stem cells. So I would advise having a quick look at that, especially in relation to this. Do not put it past OCR to sneak in stuff that's not on the content list through this kind of back door. Okay, so stem cells, have a look at the pros and cons of that. Alright guys, this has been hormonal communication. I hope you found it useful. Let me know if, because this is my version of things. If you think that, some other things could be included, if you think that the way I'm doing this could do with some modification, refinements to make them more useful, do let me know. But this has been hormonal communication, guys. The next one's going to be membranes. Good luck.